Next, 65th. Good morning, everyone. So, 59th questions here. Consider the following statement, assertion reason type of questions. The size of population for any species is not a static parameter. Reason is the density of population in a given during a given period fluctuates due to changes in four basic processes. So, we will analyze this statement. So, this questions which is from generally ecology, in which population dynamics here population dynamics. So this population dynamics which it refers to how population changes in its size or density and composition over a period of time. Okay, and these changes in over period of time, this population changes is generally due to internal and environmental changes. Due to environmental or due to internal changes only, the population will change in their size, density, and composition. Generally, these changes will occur due to four important process, four basic process. So they are first one very important. They are natality. So means birth rate. Natality means birth rate per capita birth per unit of time. Second one is mortality means death rate per capita death per unit time. And third one very important, also immigration. So if any species which is immigrate in a new habitat, we also calculate. And the fourth one emigration. So if any species from their natural habitat they are emigrate from to the another places, also we can plus. Not only these four basic process, but also there are two important factors also contribute to this population dynamics. So like sex ratio, the number of male, the percentage of male and female in a population, sex ratio. And one more important thing, that is age distribution. So this age distribution also contribute to this population changes or population dynamics. In age distribution, generally populations based on age distribution, this population, the population which is split into three important categories, like pre reproductive age group and reproductive age group and post reproductive age group, like that. So all these are the various factors or attributes or characters of population, and they only helps to the density of to know the density of population in a given period of time. So here all this. So why? Because here in first statement assertion, the size of population is any species is not a static manner. Yes. Why? Because so always the population of any species is not a static. It is always changes. Why? Because due to internal and environmental. So why it is changes means here reason is given. The density of population is a given habitat during given period 100%. They make some fluctuates due to some changes, and those changes which occur like this natality, some new species will gain birth, some new species already exited species can get death. So immigrate and emigrant, and because of their sex ratios and their age distribution, all these are the various important factors which make this population become fluctuates. So assertion and reason both are statement are true. So right answer is first one. Okay. Next one is 60th question. So it's a catadromous fish. We have to catadromous fish. So let's start some discussion about different types of fish based on their migrated. Generally, fishes migrate primarily for two reasons. Here I'm read. Fishes migrates for two reasons. One is for feeding reason, and second one is spanning reason. Spanning or breeding purpose. So here the fishes are migrate for these two reasons, be feeding and spanning or breeding from one place to another place. So based on which the fishes are categorized into different types. In that first one, very important, diadromous fish. Here diadromous fish means, so these are true migratory fish. These are true migratory fish to overcome osmotic barrier. To overcome osmotic barriers. Why? Because this diadomous fish can travel or migrate between salt water, that is marine water or sea water, to fresh or river water. So these are the true migratory fish, diadromous fish, in which these fishes can travel between salt water to fresh water only, means marine water to river water only. And in this we have two different types of fishes, like here I'm writing. And Anadromous fishes, here I have anadromous fishes and catadromous fishes. Catadromous fish. Anadromous and catadromous fish both are diadromous fish, in which remember always fishes migrate from marine water or between salt water to fresh water. So in anadromous exactly how fishes will move from where to where. So in which anadromous fishes, the fishes present the natural habitat of anadromous fishes in seawater. So they migrate from seawater to freshwater. Very important. Freshwater. The fishes which is migrate from seawater to freshwater, such type of fishes are called anadromous fish. So catadromous fish quite opposite. So in which catadromous fish, the fishes which is migrate from fresh water to marine water, means seawater or salt water or seawater. So remember, the catadromous fish always travel or migrate from fresh water to salt water. So here we have some example for the fishes which is move or travel from seawater to freshwater. For example, salmon fish and hilsa fish. Very important. Hilsa fish. This salmon and hilsa fish generally both are sea water fishes, but they are travel to the freshwater. Now coming to catadromous fish, in which freshwater to saltwater fishes migrate. Best example for catadromous fish is angula fish. Okay, so these are the different types of anadromous and catadromous fishes. So once we analyze, so here catadromous fish means the fish which travel from freshwater to saltwater. Best example is angula. So here fourth answer is right one, angula. But see here salmon and hilsa fish, and here one more we have, that is cutla fish. All these fishes are migrate from seawater to freshwater, means these are anadromous type of fish. But in question asking only catadromous fish, then the right answer is angula. Okay, next one here 61st question. Consider the following statement type questions. In statement when given, main region of digestion in stomach in human is pyloric. Lipase of bile juice play important role in digestion of fat. So this question from digestion in human, in which first we need to know stomach structures. Here stomach. So generally stomach has three important parts like this. We have no stomach. Yeah. So in stomach there are three important regions are present. So first here we have cardiac region, cardiac part of stomach, and the main body of stomach here. This body is called fundic body, fundic region, and at here we have pyloric part. So in stomach there are three important parts are there. Fundic body part, also called fundic part. Third one is pyloric part. So generally the main region of digestion in stomach occur at fundic part. So here only main digestion will occur. So not pyloric part. So where it occur generally fundic or body parts only main digestion in stomach will occur. So this statement is wrong. Now coming to lipase of bile juice play important role in digestion of fats. No, generally bile juice. So bile juice secreted from liver, we know that. So liver cells, hepatocyte cell which secrete bile juice. Remember bile juice do not contain any enzymes. Any enzymes. Just bile juice contain only bile salts. In these bile salts, in bile salt, bile juice contain only bile salt, and also which contains 97% of bile juice contain water, and also this called bile juice contain bile pigments. Only these three things are present in bile juice. No enzymes. Remember, bile juice doesn't contain any enzymes. Only bile juice contain bile salt, 97% of water, and bile pigments. Here bile salt, like we have sodium and potassium glycolate and sodium potassium tarocholate. So these are the important salts which play important role in emulsification of fat. And bile pigment, there are two important bile pigments are there. They are bilirubin and bile. So remember, here bile juice do not contain any enzyme, but here given in option lipase of bile juice. So this word is make this statement completely wrong. So why? Because lipase is an enzyme generally. Here lipase is an enzyme. This lipase enzyme which is produced from three different organs, like which is produced from the stomach, hence gastric lipase, and also it is produced from the intestine and intestinal lipase, and also it is produced from the pancreas, so pancreatic lipase. Generally these lipase are produced from various gastric and pancreatic and intestinal, but bile juice doesn't contain any lipase enzyme. So then this lipase enzyme which helps to convert fat into which make the fatty acids. But emulsification of fat, in emulsification of fat, the important role which is played by this bile juice. In bile juice, bile salt play important role. Okay, so this statement is also wrong. Why? Because lipase of bile juice doesn't contain. So lipase only present in gastric and intestinal and pancreatic. Okay, so these two statements are wrong. Hence both statement 1 and statement 2 are false. So right answer is second one. Next one, 62 question here. Reason, we have to find out reason for right side shift of oxygen dissociation curve. So this questions from which is framed from transport of oxygen here. Transport of oxygen. Generally oxygen mainly transported by two important structure by RBC. In RBC generally hemoglobin play important role to transport this oxygen, and by plasma. So 97% of oxygen transport by RBC, only 3% of plus oxygen transport by plasma by in dissolved form. Okay, generally this transport of RBC, which is mainly occur how which occur means here see. So at left side generally we know that alveoli from alveoli this oxygen which associate with hemoglobin. At alveoli oxygen is associated with hemoglobin, then it form oxyhemoglobin means here oxyhemoglobin association curve will be formed at alveoli, that is in left side. At left side. So what is the condition which favor this formation of oxyhemoglobin at left side at alveoli region means very important. Here why? Because in alveoli we have more amount of oxygen. So here partial pressure of oxygen is more. Hence which is bind with hemoglobin. So when partial pressure of oxygen is more, very important. When partial pressure of oxygen is more and partial pressure of carbon dioxide is less, and allow also temperature. Your temperature is also less and pH is also less. So these are the conditions which favor to binding of oxygen to the hemoglobin at alveoli level. Okay, at right left side. But these alveoli which from this alveoli the oxygen which bind to the hemoglobin and in the form of oxyhemoglobin the oxygen will be travel. So which travel at where? At tissue. So why? Because tissue need oxygen, no? So at tissue level means tissue means cell level, here cell. So here this oxygen association will be dissociated means they are break dissociation. So it means that at tissue level what form? At tissue level what is formed here? Oxygen dissociation will be formed. So when oxygen hemoglobin get dissociated from each other, then this oxygen, the formed oxygen will enter into this tissue. So when what is the condition? And this is make them right side at tissue level. Remember at right side here I am writing at right side at tissue level. Here the oxygen will enter into this tissue from this by oxygen hemoglobin dissociation. So what is the condition which favor to entry of oxygen means? Remember, so due to after entry here partial pressure of oxygen is must be less, but partial pressure of carbon dioxide is more and the temperature is more and pH is more. So here remember at the right side of at tissue level the oxygen and hemoglobin dissociation curve will be occur. Remember oxygen hemoglobin dissociation curve will be occur. So when means then partial pressure of oxygen is less but carbon dioxide temperature and pH is more. So this condition which favor the oxygen hemoglobin dissociation curve at left side. But when at right side, when left side, the when at alveoli level the oxygen hemoglobin association curve will be formed. Why? Because oxygen must and should be associated with the hemoglobin and transport. Okay, so here the answer is must be high pH, but in official key the answer is low pH, but this one wrong. Why? Because at high pH only the oxygen the hemoglobin dissociation curve will be occur. So right answer is fourth one, so not first one. In official key giving wrong answer. At high pH only or at high temperature at high partial pressure of carbon dioxide only this at right side at tissue level oxygen hemoglobin dissociation curve will be occur. Remember, very important. So right answer is here fourth one. So next one, 63rd question. Consider the following assertion reason type of question. In blood bank, clotting of blood is prevented by adding citrate and oxalate of sodium. Here in blood bank clotting of blood will be prevented by adding what? Citrate, citrates and oxalate of sodium. Citrate and oxalate bind to calcium ions, which prevent the activation of prothrombin. Reason. So once we analyze this statement here. So generally we know that when there is a injuries, then there is a naturally the blood coagulation or blood clotting process will occur. So here in blood bank, clotting of blood is prevented by adding citrate and oxalate of sodium. So here reason is calcium ion which prevent the activation of prothrombin. How means generally, in general, when there is an injury, blood coagulation process will occur. How means generally, in which calcium play important role. So how means generally when there is an injury, and there is one enzyme called thrombokinase enzyme which activate and which convert prothrombin into thrombin in presence of thrombokinase enzyme. And along with thrombokinase enzyme, calcium also play important role for conversion of prothrombin into thrombin. And in general, here I am explaining. And this thrombin enzyme, which again this thrombin enzyme convert fibrinogen into fibrin, and this fibrin which make this clotting of blood in general. So here calcium play important role. So when here in a blood bank, the clotting of blood is prevented by this citric and oxalate of sodium ion. So what they will do means, and these two preservatives are molecules which generally binds to the calcium. Yeah. So it means this citric and oxalate of sodium which act as a chilling agent to bind these two to the calcium ion, bind to the calcium ion. So when there is a calcium ion will bind means, it means that it remove calcium ion. These two are citric and oxalate of sodium which remove the calcium ion from the plasma completely, which remove the calcium from the plasma. From the if calcium ion which is removed from the plasma, so it means that in plasma we don't have any calcium. If there is no calcium, no thrombokinase will convert this prothrombin into thrombin. Why? Because thrombokinase need, and there is a one important factor, calcium is also important for conversion of prothrombin into thrombin. But due to absence of this calcium, no prothrombin here, no prothrombin converted into thrombin. If no thrombin, no fibrinogen converted into fibrin. If no fibrin means there is no blood clot, no clotting. So like that this process will occur. So here main reason who play important role in blood clotting means calcium. What we need to do, we need to remove this calcium from the plasma. If we need to remove this calcium from the plasma, then we have to add the citric and oxalate of sodium. Why? Because the citric and oxalate sodium which bind to this calcium and will make them calcium to become removed from this plasma. So like that no clotting will be occurring blood bank. Okay, like that we can preserve blood more time, right? So assertion and reason both are correct statement. So here right answer is both A and R are correct, the correct explanation of A. Why? Because here prothrombin, so prevent the activation of prothrombin, no prothrombin will be activated. Why? Because no calcium. Match the following type of questions. So here excretory organs are given like protonephridia, metanephridia, malpighian tubules, green glands. Here examples. So let's start some explanation on this excretory organs. So evolution of excretory organ in various phylum we will discuss. So here from phylum Protozoa to Ctenophora, there is no specialized organ. There is no specialized excretory organ or excretory system, and they perform their excretion by general body surface by diffusion, through their general body surface by diffusion only from protozoa to ctenophora. Means here protozoa to ctenophora means which include protozoa, porifera, coelenterata and ctenophora. So these four phylums, they do not have any specialized excretory organs or system, hence they perform their excretion through this general body surface by diffusion process. Now coming to after ctenophora we have phylum Platy. So in Platyhelminthes, the excretory organ is proto. So this protonephridia is also called phlegm cell. Phlegm cells. Or also we can use another term solenocyte. Solenocytes. So in which Platyhelminthes perform excretory system. And coming to after Platyhelminthes we have Aschelminthus phylum. Aschelminthus means here nematoda phylum. So in nematoda the excretory organ here nematoda renate cell. Renate cells. So through this renate cell they perform excretion. And after Aschelminthus we have Annelida. Include earthworms, leech like that. Annelida. So in Annelida, the excretory organ is nephridia. So very important. Coming after Annelida, we have Arthropoda phylum. Here I am writing Arthropoda. So here the Arthropoda phylum has, and it is the largest phylum in animal kingdom. Remember, and this Arthropoda has three important excretory organs. So they are Malpighian tubules. One is Malpighian tubule, and one more is coxal gland, and one more is green gland, also called antennal gland. Green gland, also called antennal gland. So these are the three important excretory organs are present in arthropoda. So generally these Malpighian tubules are seen in terrestrial arthropoda organism. Terrestrial arthropods organisms. Terrestrial arthropods means which include insects. So insects generally we have seen Malpighian tubule as a excretory organ. Now coming to coxal gland. So coxal gland are seen in arachnida class in arthropoda. So arthropoda has various classes. In that arachnida is a one type of class. This arachnida class of arthropoda having this coxal gland as a excretory organ. So generally arachnida they include like spiders and scorpion. Spiders and scorpion. So these are the various organs including arachnida, and it has coxal gland as a excretory organ. Now coming to green gland or antennal glands, which is generally seen in aquatic arthropoda. Aquatic arthropods. Here aquatic arthropods include very important like crustacean class in arthropoda phylum. So this crustacean class which include a very important organism like prawns. So prawns is belongs to crustacean class which belongs to arthropoda phylum, and it has aquatic excretory organ, that is green gland or antennal gland. So these are the various glands are seen in arthropoda. Now after arthropoda we have phylum called Echinodermata phylum. So generally very important Echinodermata phylum. It doesn't sorry, not Echinodermata. Okay, we will discuss. Not after arthropoda we have mollusca. Here I am writing mollusca phylum. So mollusca phylum is the second phylum in animal kingdom, but arthropoda is the first largest phylum. Remember, first largest phylum. So generally in mollusca, the excretory organs of bogens. Organs of bogens are the excretory organ in mollusca. So then in echinodermata, very important. So in generally this echinodermata, it does not have any specialized, no specialized excretory organ, but they perform their excretion by tube feed. Very important. So even though echinodermata does not have any excretory organs, but they perform their excretion through this tube feed. So then after coming to here next, after echinodermata we have hemichordates. So hemichordate phylum we will discuss. So in hemichordate phylum, here in hemichordates the excretory organ is proboscis gland, the excretory organ in hemichordate. Now coming to very very important vertebrate. In vertebrate the excretory organ is kidney. In all vertebrates the excretory organ is kidney, but based on the location of kidney we have different types of kidneys. If kidneys are located in head region, such type of kidney is called pronephridian kidney. If kidney located in thoracic region, thoracic means lung region, such type of kidney is called meta kidney. If kidneys are located in abdominal cavity or abdominal region, such type of kidneys are called meta. Yeah, here meso, sorry, not meta. Thoracic contain meso, but here abdominal having meta kidney. So metanephridian kidney means the kidney which is located in abdominal region. Meso means thoracic region. And pronephridian kidney located in head region. So generally these pronephridian kidneys are generally seen in fishes, like which is generally seen in two important organisms like cyclostomes, and also which is seen in tadpole larva of fishes or amphibians. Okay, so in like cyclostomes and tadpole larva we have seen which type of kidney means pronephridian kidney. Now coming to thoracics, like in region called mesonephridian. These mesonephridian kidneys are generally seen in two organisms. Very important, they are fishes and amphibians. In fishes and amphibians we have seen mesonephridian kidney, means in which the kidneys are located in thoracic region. A very important abdominal kidney which is generally seen in reptiles and Aves are nothing but birds and very important mammals are include human being. So in these three classes we have seen abdominal kidney like called metanephridian kidney. So in this organism we have seen metanephridian kidney. So all these are the evolution of excretory organs in various phylums we discuss. Now here we analyze this MCQs here. Protonephridian kidney. Here don't confuse, proto is completely different from pro. So pronephridian kidneys are seen in cyclostomes and tadpole. But here asking protonephridian kidney also known as phlegm cell. Phlegm cells are also called solenoid cells, which is generally seen in which organism is platyhelminthes. So here we have to identify the platyhelminthes are also which is seen in larva annelids. We have seen this type of kidney. Okay, so here no platyhelminthes, but here we have larva of annelids. So right answer A is third. Now coming to metanephridian kidney. Here metanephridian kidney. So these metanephridian kidneys are generally seen in annelids. Okay, so here nephridia means also in human being also we have metanephridian kidney, but here metanephridia or nephridia, so which is seen in annelids. So here fifth one, Malpighian tubules generally seen in terrestrial arthropods. Best example is insects. So C is one. Green gland, just now we discussed green glands are seen in crustacean. It is aquatic arthropods. Or green gland also known as antennal glands. So here D1, D is 2. So here option third, 4, 1, 2. We have to choose. Yeah, third here. Third, 4, 1, 2. So right answer is second one. 6, 5 questions. Study of the following and choose the correct statement. Here we have to choose correct statement among 4 statements are given. So in which first sarcoplasmic reticulum is the storehouse of the calcium ion. Thin filament. Here giving of myofibril is formed by actin, troponin, myosin molecules. Thick filament of myofibril are held together by M line. The portion of myofibril between two successive M line, if you are giving M line, is called sarcomas. Okay, so these are the few options are given. Let's start some analysis of these questions. Here, these questions which completely came from muscle. So here we know that hierarchy, structural hierarchy of muscle, structural hierarchy of muscle. Generally, for example, if you have like this muscle structure, now think it is muscle, and this muscle which covered with a layer, the layer which is present around muscle like this. So here this muscles is called, this layer is called AP. Okay, now these muscles which contain bundles like muscle bundles like this. Okay, so these are muscle bundles are also called fascicles. Now these fascicles are muscle bundles also enveloped with a layer. So around each muscle bundles we have a layers like this, and this layer is called peri, and each muscle bundles contain group of here like this. Sorry, here group of various structures are called muscle fiber. So this is what muscle fiber. So here muscle fiber is nothing but muscle cell, and these individual muscle cells also surrounded by like this. See, this is muscle cell now. So this muscle cell also surrounded by a individual muscle fiber surrounded by a layer is called endomysia. This muscle fiber is a muscle cell, and this muscle cell, if you see the internal anatomy of muscle fiber or muscle cell, and it has like this structure. For example, this think it is a muscle fiber or muscle cell, and these muscle cells contain various structures as like normal typical cell structures, and it contains its own plasma membrane. And this plasma membrane is called sarcolemma, and it contains its own cytoplasm called it contains its own mitochondria called sarcosome. It contains its own endoplasmic reticulum like this called sarcoplasmic reticulum. And remember, this muscle cells contain multinucleated. So here we have multinucleated. So in multinucleated condition, the nucleus is called nuclei. So here multinucleated. So these are the internal structure of muscle fiber or muscle cell. But here we need to know, here this sarco means the cytoplasm of the muscle is called sarcoplasm. Now this sarcoplasm contain a specialized cell organelles. Here I am writing. The sarcoplasm contain a specialized cell organelles. So that cell organelle is nothing but myofibril. Very important. So myofibril is a specialized cell organelle which is present inside the sarcoplasm of muscle fiber. And do you know this myofibril, which is like how they are like present means here in this generally this myofibril are present inside the sarcoplasm of the muscle fiber like this. So they are long and thread-like structures. Okay, there are hundred to thousands of myofibrils present parallel to each other. So myofibrils appear like it's a long and thread-like structures, and there are hundreds to thousands of myofibrils present in sarcoplasm of the muscle fiber, like parallel to each other. They are parallel to each other like that. There are hundred to thousand of myofibrils are present in the sarcoplasm. And a very important thing here, each myofibril, see, think it is a myofibril, single myofibril. Each myofibril is made up of with filaments. Those filaments are called myofilament. Myofilaments. Each myofibril made up of with the filaments called myofilaments. Generally, there are two filaments are present. One is thin filament, another one is thick filament. Thin and thick filament. Remember, thin and thick filament. First, thick, thin, and thick filament. Here, the thin filament is called actin. These thin filament are made up of with actin. This thick filament is made up of with myosin. Remember, thin filament is made up of with actin. Thick filament is made up of with myosin. Okay. So along with actin, there are some other contractile protein also help to made up of this thin filament, like troponin and tropomyosin. So these are the various filaments are present and which all together made the thin filament in myo, but thick filament is made up of with only myosin. And remember, if you see the structure of this myofibril, they are like, so think it is a one type of only one myofibril structures. So these myofibrils are made up of with thin filament like this. So this thin filament is nothing but actin protein, and also which is made up of with thick filament like this. So in between here we have, now here in between thin filament we have thick filament. So here this thin filament is made up of with actin protein, and this thick filament is made up of with myosin. Like that, the internal structure of filaments are present inside the each myo. Okay, and here so we have different types of lines and zones. Exactly in between here, in between this M line is present exactly middle of the thick filament. Here we have M line, and there are Z line is present here between Z line, between exact present between the thick and thin filament, means like this. Here we have Z line. See, this is what Z line. So here two successive Z line, like that we have different Z lines, two successive Z line. There is a gap, and this structure is called sarcomer. So this sarcomer is the structural and functional unit of the muscle. Structural and functional unit of the muscle is sarcomer. Okay, so remember all this concept with related to the muscle fibers, cells or myofibril structures. So let's analyze this questions here. See here, first statement here given, sarcoplasmic reticulum is the storehouse of the calcium ion. Yes. So here, sarcoplasmic reticulum is a type of cell organelle present in muscle fiber which store mainly calcium ion. So this statement is right. Thin filament. Here giving thin filament of myofibril is formed by, yes, actin, troponin, myosin molecules. But here say this myosin, no. Myosin is a molecule of form thick filament. Okay, thick filament. But here in questions asking only thin filament of myofibril is formed by actin, troponin, and also tropo. But here giving myosin is a protein molecule which is formed the thick filament. So this statement is wrong. Thick filament, here giving thick filament means the filament which is made up of with myosin protein. This thick filament of myofibrils are held together by M line. Yes. So here this thick filament are held together by M line. So this statement is right. The portion of myofibril between two successive, here this is the myofibril structure. So in which there are two successive Z line, not M line. Here sarcomer means the portion of myofibril between two successive Z line. Here Z line is known as sarcomer, and sarcomer is the only structure which is structural and functional unit of the muscle contraction. So here M line is not. Only Z line is a successive portion between myofibril is known to be as a sarcomer. So this statement is wrong. So here first and third statement is correct. First and third. See here. So here first and third statement is make right statement. Next one, photopigment visual purpose purple is present in. Okay, so here giving rods, cones, choroid and iris. These are different types of layers in eyes. So let's start discussion on photosynthetic pigment in retina. About eye structure, innermost layer in eye is called retina. Innermost layer of eye is retina. This retina has photo cells. So there are two photoreceptor cells are present in retina. One is rods, another one is cones. Here rods, very important, it shows scotopic vision. Here scotopic vision means night vision, a dim vision, night vision, and cones shows photopic vision. The photopic vision means daylight, day vision, daylight vision. Here important thing here, these rods contain only one pigment in rods. So that pigment is called rhodospin, very important. So rhodospin is a pigment present in rod cell. It is a photo cell in innermost layer of eye, that is retina, rhodospin. And this rhodospin also we can use another term purple color, visual purple pigment. Also we can use another term visual purple pigments. And here this generally this rhodospin pigment, which is a derivative of vitamin A. Important here, it is a derivative of vitamin A. So here vitamin A, vitamin A is nothing but retinol. So rhodospin is a derivative of retinol, from which rhodospin is derived, and it is has a also known as visual purple pigments. Okay. Now coming to cones, generally cones having three different types of pigments. In that, one is chloropsin. So chloraspin, chloro means green, so it gives green color. And next one is cyanopsin, cyano means blue, so it gives blue color. And one more is there, that is erythropsin. So erythropsin gives red color. So like that, various colors are giving due to various pigments are present in these cone cells. So like that, G, green, blue and red, so these are considered as a color primary color. Do you know there is a night blindness or color blindness? So here color blindness is due to any one of the deficiency of pigments, any one of the deficiency of this pigment, cone pigment. Remember, any one of the deficiency of cone cells or cone pigment, it leads to cause color blindness. And we have one more blindness, that is night blindness. So this night blindness is due to deficiency of vitamin A. So vitamin A deficiency leads to cause night blindness, and color blindness is due to deficiency of any one of the cone cells or cone pigments. So this is all about the important analysis on internal structure of eye, that is retina. So here in question, the photopigment visual purple is present in means in which rod? Why? Because a visual purple color also we can use rhodospin. So rhodospin pigment present in rod cells only. So the right answer is rod. Yeah. So next one, 67 question here, giving hormones and endocrine glands. The respect to endocrine gland, we have to correct this answer. Match the here giving various hormone like vasopressin hormone, aldosterone hormone, secretin hormone, calcitonin hormone, and their respective parts from which we obtain these hormones. Okay, so let's start some explanation on vasopressin hormone here, about vaso hormone. So generally, vasopressin hormone, also known as ADH hormone, antidiuretic hormone, antidiuretic hormone. So here generally this vasopressin hormone synthesized in but they transport and store at posterior pituitary gland. Very important. Generally, these vasopressin hormones are synthesized in hypothalamus only. See, so if you think this is the hypothalamus, and here we have pituitary glands. This is what hypothalamus. This is pituitary. In pituitary, we have two parts: anterior pituitary and posterior pituitary. But the synthesis of vasopressin, not only vasopressin, along with vasopressin, oxytocin hormones also synthesized in this region, in hypothalamus region, but they transport to the posterior pituitary. Here only they will store. Okay, right. And very important here. So the function of vasopressin is which helps to water reabsorption from the region of density and collecting duct of the nephron. So from nephron, density and collecting duct region, there is a absorption of water in presence of this vasopressin hormone. So when there is a reabsorption of water will occur, then it prevent diuresis. The diuresis means to prevent excess loss of water through urine. Prevent excess loss of water through urine. Okay. So not only that, vasopressin also act as a vasoconstrictor. Very important. Vasoconstrictor. But aldosterone act as a vasodilator. But this one act as a vasoconstrictor. Means contraction of blood vessel. Vasoconstriction means contraction of blood vessels. So when there is a contraction of blood vessel will occur, so definitely there is an increase of blood pressure and glomerular filtration rate. Both are increased due to vasoconstrictions. Okay. So these are the various important notes on vasopressin. So finally, vasopressin are synthesized from duodenum, pituitary gland, adrenal gland, thyroid gland, or thymus gland. Here the right answer is pituitary gland. Here, so vasopressin is related to pituitary gland. Here storage. Okay, in pituitary, which pituitary gland means that is posterior pituitary gland. Remember. Now coming to aldosterone. So here this aldosterone is a type of mineral hormone synthesized from adrenal cortex. Adrenal cortex of adrenal gland is mineralocorticoid, that is aldosterone hormones are synthesized. Okay, and here the main role of this the target site is DCT and collecting tube of nephron only. So they target only DCT and collecting tube like vasopressin, but they play important role like they play important role here. Yeah, so they play important role. Aldosterone play important role in different functions like it electrolyte balance, helps in electrolyte making electrolyte balance. So it means electrolyte balance means it conserve sodium and eliminate the or remove the high excess potassium. So conserve sodium and eliminate the excess potassium. If there is excess potassium in blood and eliminated by this aldosterone, but there is a very important, there is a need to conserve the sodium. Not only electrolyte balance but also helps in body fluid value, to maintain body fluid value. Okay, and also maintain osmotic pressure of blood, and very important, it also helps in maintain the blood pressure, BP, and GFR, glomerular filtration rate. So these are the various functions of aldosterone. So remember, the main target site is DCT and collecting tube, and mineralocorticoid is a which is synthesis from the adrenal cortex of adrenal gland. So here we have to choose where adrenal gland here. So third. So first here, second, this one second, this one third. And now coming to secretin. About secretin. Next. About secretin. So here secretin, it is a GIT hormone, gastrointestinal hormone, which is secreted from duodenum of small intestine. Remember, do you know the secretin is a first discovered hormone. It is a first discovered hormone. It was discovered by two scientists, Bayliss and Starling in 1902. Remember, Bayliss and Starling. Here, secretin target organ is exocrine part of pancreas. Target organ. The target organ of secretin is exocrine part of pancreas and helps to synthesis of water and bicarbonate ion from pancreas. So here secretin helps secreted from duodenum. So here first one. Just now we discussed about aldosterone is a third one, and vasopressin second one. Now coming to calcitonin. Here I am explaining calcitonin. About calcitonin. So here calcitonin are also called thyro-TCT. So generally it is a protein or peptide hormone. It is a protein or peptide hormone. Unlike T3 and T4. Generally T3 and T4 hormones of iodized tyrosine derivatives. They are iodized tyrosine is a amino acid tyrosine derivatives, T3 and T4 hormones of thyroid gland. So this calcitonin secreted from this thyroid gland. Remember. So thyroid gland secreted generally T3, T4 and TCT, means thyrocalcitonin hormone. But among which TCT is the only peptide or protein hormone, but T3 and T4 are the iodized tyrosine derivative hormones. Remember, and you know this thyroid gland which mainly secreted to these hormones, from two different types of cells. This thyroid gland has follicular cell, and also it has parapellicular cells. Para cell. Now this parapellicular cell also called as C cell. Now these parapellicular or C cell secrete TCT hormone, thyrocalcitonin hormone. But this pellicular cell secrete this T3 and T4 hormone. Remember. Okay. So like that, what is the main role of this calcitonin means, which is very important to regulate blood. So blood calcium, very important. Calcium, blood calcium regulation hormone it is. Blood calcium regulation hormone is calcitonin. So in name only it is giving calc. Calc means calcium in blood. Okay. So in blood it maintain the calcium regulation. So when means and hence this calcitonin hormones also known to be as a hypocalcemic hormone. Hypocalcemic hormone, why? Because which lower the blood calcium level when there is a excess calcium in blood, which lower the calcium level in blood. If there is a excess calcium level in blood, then which this thyrocalcitonin hormone which regulate and which lower the calcium level in blood, it means that and it send the excess calcium to the bone. Okay. So like that it perform as a hypocalcemic hormone. So these all are the various function of calcitonin hormone which is secreted from the parapellicular cells of thyroid gland. So here calcitonin is a thyroid gland. So here let's start. Yes, here vasopressin secreted from the pituitary gland. Which pituitary gland? Posterior pituitary gland. Second one, aldosterone from adrenal gland. In which part of the adrenal gland? Cortex part of the adrenal gland. Hence third one, secretin is a gastrointestinal hormone secreted from the duodenum. Hence first, calcitonin from the thyroid glands. Hence fourth one. So here two, third, one, two. So right answer is first one.